Welcome class. In this session, we'll be discussing regarding the two phenomena, that is photoperiodism as well as vernalization. So first we'll look into photoperiodism. So this phenomenon was first studied by two scientists, that is Gardner as well as Ellard. So these two first conducted studies related to photoperiodism and the effect of daily duration of the light hours as well as dark periods on the growth and development of the plants, especially flowering, is called as photoperiodism. So this is the definition of photoperiodism. In simple, we can say the effect of the duration of the light and dark hours, okay, in the daytime in a 24 hours period on flowering of the plant, that can be considered as a photoperiodism. Not only it affects the flowering, it also affects the other growth and developmental pattern in case of plants such as stem elongation and all. So in total, we can say that the effect of daily duration of light hours and the dark periods on the growth and development of plants, especially flowering, that can be defined as photoperiodism. And here, the relative length of the day and the night period, that can be considered as a photoperiod. And in response to photoperiod, the flowering will be induced in case of plants, and this phenomenon is photoperiodism. So that is a simple definition. And in case of photoperiodism, so specific chemical will be involved in this event, and the chemical that is going to perceive the photoperiodic stimulus, that is called as phytochrome. And this phytochrome is a type of a chromoprotein which is present in a cytoplasm, and it is blue colored, and generally this particular phytochrome, it will be present in the leaves, so that during flowering, the stimulus for the flowering in case of plants, it will be perceived by the chemicals present in the leaves. As a result, the leaf bud will be modified into flower bud, and the flowering will be induced in case of the plants. And here, the length of the day and night above and below which the plant cannot flower, so that particular time period, it is called as critical photoperiod. Suppose if we consider a day length of 24 hours. So in case of, this is the 24 hours day length here. So if we consider in this, so let us say 12 hours is for the light period and 12 hours is for the night period. Okay. So here the critical photo period means, so here it is a duration. So let us say almost up to 8 to 10 hours of minimum light duration is required for the plants to induce flowering. Suppose if the plant is unable to produce the flower below the minimum day and night length or the night period that is available, so that particular period, it is called as critical photo period, or length of the day and night. Above also the plant cannot produce flowering, or below also the plant cannot produce flowering. So that particular period, that is called as critical photo period. So this critical photo period, it varies depending upon the plant and all. So in general, we can say it is a length of day and night above and below which the plants fail to flower. So that is called as critical photo period. And based on the photoperiodic responses, plants can be categorized into three different categories, such as long day plants, short day plants, as well as the day neutral plants. So in case of long day plants, so these plants, they require a light hour above the critical length, then only they can induce flowering. So such plants are called as long day plants. Suppose in a particular day, in a 24 hours, if we consider almost, so these plants require 16 hours of the light exposure to in a minimum context. If we consider 16 hours of the light exposure is required by such plants. So such plants are called as long day plants, where they require relatively longer light hours which are above the critical length of the length for the flowering. So examples are spinach, wheat, henbane, potato. So all these are examples for the long day plants. And coming to the short day plants. So these are the plants which require photoperiod less than the critical day length. Suppose if we consider 24 hours of the 24 hours length of the day. So here these plants, they require less than 8 hours of the day length. Okay, so such plants, the flowering can be induced in case of the short day plants if the day length is up to 8 hours or less than the 8 hours. So such plants are called as short day plants, where they require photoperiod less than the critical day length for the flowering. So examples for such short day plants are tobacco, soya bean, as well as the xanthium. And for the first time, the photoperiodism has been studied in case of a variety of a tobacco only. So where they observed when this tobacco has been cultivated in a different areas or the different habitats, so their flowering pattern has been changed depending upon the available day length. So based on that, the Allard as well as so these two scientists such as Garner and Allard, so they gave a concept of photoperiodism. So examples for short day plants are tobacco, soya bean, as well as the xanthium. And next one is the day neutral plant. So whatever may be the day length, so it is not going to affect the flowering period in case of the plants. So these are the plants that are insensitive to the day length and flowers at about the same time under all the day lengths. So such plants are called as day neutral plant. They are not sensitive to this particular day length, whether it is maximum or minimum, it is not going to bother their flowering. So at particular time only it will be flowering in all day length condition. So such plants are day neutral plants. Examples are cucumber, cotton, as well as the pea. So these are some examples for the day neutral plants. So next, if we see the mechanism of photoperiodism. So in case of the photoperiodism, so this phytochrome that is present, so it will be sensitive to the two lights and it exhibits in a two subtypes such as PFR as well as the PR forms. So this PFR is, it can absorb the far red light region. PR can absorb the red light. So in response to far red as well as red light, so this concentration of this PFR and PR form in case of plants, it induces flowering in case of long day as well as the short day plants. Suppose if we consider in case of short day plants, so or we can consider them as a long night plants. So they will flower when the night exceeds a critical dark period. A flash of light interrupting the dark period will be preventing the flowering. And generally what happens here is actually, so here in the darkness, so what happens? So this PFR form will be converted back to the PR form during the darkness. Okay. And if the dark period is prolonged, so concentration of the PR form will be more and the less concentration of the PFR form will be there. So if concentration of PFR form of the phytochrome falls in case of short day plants and PR form exceeds, then what happens? It induces flowering in case of the short day plant. So this PR form, more concentration of PR form in case of short day plants, it induces flowering in them. Okay, and this PFR form will be converted to PR form during the darkness. In the night period, it will be converted. Whereas in the daytime, this PR form will be converted back to the PFR form. So that's why in case of long day plant, what happens? So they will be requiring a short night period or the long day period. So in such plants, what happens? So a brief flash of a light artificially interrupts a long break period, thus it can induce the flowering. So in case of the short day plants also, if you introduce a short flash of light, so it prevents flowering because the long dark period is inhibited. Similarly, in case of long day plants, suppose if it is a long dark period also, it is a short day period also, if you artificially introduce a flash of light, immediately it can induce flowering in case of such plants. So that is because In case of long day plants, what happens? So here the PFR will be converted back to PR during the night time, but as the night duration is lesser, so PFR concentration will be more in case of the plant, okay, and increase in the PFR concentration in case of long day plants. So increase in the PFR form, so it will induce flowering in case of the short day plant. So that is the mechanism that is lying behind the photoperiodism in case of plants, where the phytochromes, they exist in two forms, PFR and PR form. PFR concentration will be more in the daytime and PR will be more in the nighttime. So if PFR concentration is more, so in case of long day plants, it induces flowering, whereas PR concentration will be more in case of the short day plants where it induces flowering. Suppose if flowering period is interrupted, in case of short day plants, flash of light can prevent flowering, because immediately, so here it is PFR. So PFR will be converted to PR during the night period. So if we introduce the flash of light, again this PR will be converted to PFR form, and further it will be slowly converted to PR form. So the concentration of PR form will be falling in case of the short day plants, so it won't induce flowering. Whereas in case of long day plants, if you see what happens, so this PFR form will be more in concentration and slowly it will be converted to PR form during the night time. So in this case, concentration of PR form will be more because dark period is more, so hence there will be no flowering. Whereas in third case, if you see a flash of light, it immediately converts the PR form into PFR. So as a result, so this PFR concentration increases again so that it can induce the flowering in case of the plant. So that is regarding the mechanism of photoperiodism that can be seen, which will be dependent upon the concentration of PFR as well as the PR form. So next is regarding vernalization. So vernalization, it can be defined as a process of shortening of juvenile or the vegetative phase and hastening flowering by a precocious cold treatment. So here in simple we can say it is inducing the flowering in case of the plants, okay, upon cold treatment. So that is called as vernalization, where in this process the vegetative phase will be shortened and flowering period will be fastened, or it will be induced earlier by the precocious cold treatment. So this helps in transformation of winter varieties into spring varieties, and vernalization can also be done in case of apical as well as all the meristematic cells, where it will speed up it will minimizes the duration of the vegetative phase and it will prepones the flowering period in case of the plant. So this vernalization can be exhibited in case of plants such as wheat, cabbage, European rye, even in case of radish also this vernalization can be done. So in case of wheat, if we see, so there are two varieties, whereas winter as well as the spring varieties. So this winter varieties, they are harvested in the mid-summer region, whereas the spring varieties, they will be available for harvesting in a next spring season. So that is regarding these two varieties. Suppose if this winter variety has to be converted into a spring variety, so if the spring variety has to be harvested in the winter season or in the midsummer season only, so what we can do, the spring variety can be given a cold treatment so that the conditions of the variety, the conditions will be recreated, artificially created, so that it is experiencing the cold season, so that the spring variety will be converted to winter variety, and in the next season, immediate season itself, it will be ready for harvesting. So that is how the flowering period can be preponed in case of such plants. So here we can see the diagrammatic representation. So it is a slightly germinated seed. So if it is chilled for a month at 5 degrees Celsius, or it is given a cold treatment of 5 degrees Celsius, and when it is planted at ordinary temperature, so it induces flowering very quickly in case of such plants, whereas in case of the untreated plant or the non-vernalized plant, if we see, still they will be in the vegetative phase itself. So that is one benefit of the vernalization, where the yield can be, or the yielding period or the reproductive phase of the particular plant can be preponed upon cold treatment. So that is regarding vernalization. So followed by that, there is also one more concept called as seed dormancy. So seed dormancy is the phenomenon where the seed fails to germinate. So seed fails to germinate into a seedling. So that is called as seed dormancy. And this seed dormancy, it is mainly induced by the abscisic acid. Even ethylene can also induce the seed dormancy. Apart from that, the environmental conditions also, it may induce the seed dormancy, or the nature of the seed coat. So if seed coat is thick and resistant, it can resistant to the moisture. So then also it can exhibit seed dormancy. So where the seed fails to germinate to produce the new plant. And this seed dormancy can also be broken either by the application of the gibberellic acid, or by treating the seeds, by subjecting the seeds for the chemical treatment, such as treating them with the mild acids such as nitric acid. Similarly, we can also treat with the sulfuric acid. And then apart from that, we can also give a cold treatment for such seeds, where they will be chilled in a cold temperature, and this process is called as stratification. Whereas we can also go for the scarification, where the physical rupturing can be done on the seed coat. So physically we can rupture or break the seed coat so that it can be made to, the embryo can made to absorb the water, thus it favors the germination. So this is how we can break the seed dormancy, and that is regarding the phenomenon called as seed dormancy. So that is regarding the synopsis for this particular chapter, plant growth as well as development.